Very much indeed, Chair, and Felici and the organising committee, thank you so much for inviting me to give this talk. My title, actually, I thought I would be the one that was stressed at the end of the day, so I've changed the title a little bit, and I'm plagiarising from a good friend of, I think, many of the people here, John Thunder. I asked John to give a talk at a meeting we had in Perth, Australia recently, and John said, sure, I'd love to do that, I'm just going to call it Reflections. And actually what I do want to do is just to reflect a little bit and reflect about preterm birth and acknowledge Steve Lai, Al, and Al Balking Felici as wonderful colleagues, and a very talented PhD student, Grace Yang. I'll show you just a little bit of her recent data. Well, let me remind you that preterm birth is a global problem. 13 million babies are born preterm each year. There's enormous variation in different parts of the world in preterm birth rates, from 2-3% perhaps in some parts up to 15-20% in other parts of the world. The preterm birth contributes more to under 5 deaths in AIDS, malaria or tuberculosis. Than a million babies around the world die within a month or so of birth, and survivors experience long-term problems including cerebral palsy, visual, hearing and neurologic impairment. So this really is a very, very major and important area. And despite the investment of substantial funding from NIH and other bodies, we've really not made a lot of progress in reducing the overall preterm birth rates. These are the figures from the United States running at around 12% in this particular analysis. Figures from Australia, where I'm spending some time at the moment, a little bit lower overall, but you can see really not very much progress at around about 8% or so preterm birth rates for the country as a whole. And the problem perhaps is that we have been lumpers rather than reducers in looking at these sorts of data, and that's why I wanted to reflect just a little bit, because there's some really very obvious differences that we need to take into account that we haven't. These are data for British Columbia, the data for Canada, the data for other parts of the world are very similar, and what they show you very clearly is that preterm birth rates in pregnancies carrying a male fetus are about 1% or so higher than in pregnancies carrying a female fetus. So it would make sense that as we look at diagnostic measures for recognizing preterm birth rates, that we might take fetal sex into account. It's a fairly obvious one to do. And it also begins to point to the fact that we need to take the gestational age into account, because the incidence of male over female predominance is much higher earlier in gestation. This series of studies that was published 10, 12 years or so ago. And then, so more males than females born here, and then as you get closer to term, we get closer to a birth ratio of unity. And we also have to take into account ethnic differences. Now, it's not just take into account. It seems to me that trying to understand the causes of some of these differences may be actually quite valuable in understanding the underlying mechanisms of this process. John Newnham from Perth, Australia, showed that the preterm birth rates in China, in Jiangsu province, in rural and urban centres, were running at about 2.6, 2.9%. And initially we said, well, maybe there's some reporting issues there. You need to look at that differently. So John looked at preterm birth rates of women and showed out differences in country of birth and declared ethnicity, but all within Western Australia. And women born in China who needed an interpreter, and the interpretation of that is that they're relatively recent immigrants to the country, Southeast Asian, 2.5%, pretty much the same as the preterm birth rates of women in China. China, without an interpreter, born in China, without an interpreter, so perhaps more integrated, creeping up now about 5%, Australians, Caucasian about 8%, Australians, Southeast Asians about 9%, and Australians of Aboriginal descent about 15 or so percent. It seems to me that understanding perhaps the differences in preterm birth rates of Chinese women in these different circumstances, almost certainly there has to be some environmental epigenetic influence, would be extremely useful in just understanding what preterm birth rates really mean overall. And the last point I just want to make here is that the increase we see in preterm birth rates, certainly in Western Australia, and I think these data would certainly be true in Canada and in the United Kingdom as well. It's in the 32 to 36 week groupings that we're seeing the increase, that the early preterm births have remained pretty much constant over the years. And so the point is that of these early slides and this reflective part is that preterm birth is a complex syndrome with different etiologies, and if we don't understand those etiologies, it makes it pretty difficult to diagnose and treat the patient. That we might think of late preterm birth as being classic stress, uterine overdistension, bleeding, perhaps, an earlier preterm birth much more associated with an underlying infective process, perhaps cervical insufficiency. The difficulty and the problem, it seems to me, looking at the literature, is preterm birth in most trials is simply defined as a single endpoint, regardless of epidemiology, for epidemiologic purposes, regardless of etiology, and that's led to uniformly largely unsuccessful interventions for preterm birth. Well, perhaps that's not surprising because you're using one bullet to treat a whole range of different diseases. It's rather like, and I love this sort of tongue-in slide that people in this field have been, the driving force of the individual has been the desire to find their own particular golden nugget. The more facts we learn, the less we understand. And it's been a little bit like the Chinese saying it's difficult to find a black cat in a dark room, especially if there's no cat. So I wanted just to throw that in. It's late in the day and I want people just to pay attention. So let's just consider the different pathways to preterm birth that we and Felici and Charlie Locke and many others have looked at, and really there's two stress pathways up here: activation of the maternal fetal HP axis, inflammation and infection, and I'll show you, and we've already talked about this today, the very close overlap interrelationship between those: decidual hemorrhage, a thrombotic pathway, and pathologic uterine distension, with different effector molecules resulting in decidual and fetal membrane activation, upregulation of matrix metalloproteinases, prostaglandins, leading to cervical ripening, rupture of the membranes, and an epithelial mesenchymal transition perhaps taking place at that point, promotion of uterine contractions, and inappropriately preterm birth. And of course, what that means is that there's just a bunch of different potential treatments that you can use in the management of preterm birth, and if ever there was a condition that calls out for precision personalized medicine, it surely has to be this, and this whole area has to move very rapidly, it seems to me, into that area. Let me link together then the stress pathways with the nutritional pathways and talk just about where glucocorticoids come from in pregnancy, and particularly where fetal glucocorticoids come from, because of course there's a rise in fetal glucocorticoids in late gestation as a normal part of the maturational process of late gestation, but during pregnancy, maternal stress, maternal undernutrition may give rise to increases in maternal glucocorticoids, which can cross the placenta into the fetus, and will do so particularly if this placental 11-beta-hydroxysteroid dehydrogenase barrier that normally metabolizes maternal cortisol into inactive cortisone, if that barrier is broken down. In those circumstances, you would expect there to be more maternal cortisol passing into the fetus. Maternal undernutrition is one of the factors that contributes in turn to fetal undernutrition, fetal hypoxemia. You get to those same endpoints with reductions in uteroplacental blood flow, with placental insufficiency, with reductions in cord blood flow, for example, a nuchal cord, or in circumstances of high altitude where the adaptation to that high altitude may not have occurred. And in these situations, collectively, we see upregulation of fetal hypothalamic pituitary adrenal function, upregulation of appropriate genes, CRH, POMC, and steroidogenic enzymes, with increased fetal adrenal cortical activity and increased glucocorticoid concentrations in the fetal circulation. And let me just link together the stress and the undernutrition pathways with this study that we published 10, 12 years or so ago now with Peter Gluckman and Frank Bloomfield, where we were looking at the effects of periconceptual undernutrition in the sheep. Animals undernourished for 60 days before and the first 30 days of gestation, and to our surprise, when we looked at gestation length on your right, with the Kaplan-Meier plots, here's the normal lengths of gestation, around about 145 days, and you can see what happens in the periconceptually undernourished animals, the paler line. There's about 50% of those animals actually delivered prematurely, precociously. And when you look at the prepartum changes in glucocorticoid, cortisol concentrations in the circulation of these chronically catheterized fetal lambs, now another set of fetuses. And what you see is that the normal increase before term occurs with the same time as the undernourished animals who deliver at term, but that there's a number of those animals, about a half of them, who deliver prematurely. And those animals have upregulation of CRH expression in the hypothalamus, of POMC in the pituitary, interestingly, particularly in the pars intermedia, and the 17-hydroxylase and 3-beta-hydroxysteroidehydrogenase in the fetal adrenal cortex. Those pregnancies also have changes in 11-beta-HSD expression. And it's a study that Kristen Connor carried out. Remember, the animals are undernourished 60 days before conception for the first 30 days of gestation. We then looked at the placental tissue at day 50, 85, 120, 131, terms out here at 145 days. And looking at the 11-beta-HSD2, and I'm just showing you activity here, we have the same data per messenger RNA, and you can see reductions in 11-beta-HSD as a consequence of periconceptional undernutrition at day 50, at day 85, and at day 120. We've lost that effect by day 131. What that means is when you look at the ratio of cortisol F to cortisone E in the fetal circulation, in the fetuses of undernourished mothers, the cortisol to cortisone ratio is significantly higher in that early stage of pregnancy at day 50, at day 85, and we just start to lose that effect by day 120. Why is that significant? Because if you give very small amounts of glucocorticoids to the fetus by administration of synthetic glucocorticoids to the mother in early gestation, as Torsten Brunner and others have shown in our lab, you can induce programming of the HP axis and produce precocious activation of the HP axis, enhanced responsiveness in the latter part of gestation. What's really interesting comes out from these experiments. If you then take the animals through to term and start to look at changes in hypothalamic glucocorticoid expression, and we've looked here at promoter methylation, DNA methyltransferase activity, and then mRNA expression in singleton animals and in twin pregnancies, controls and underfed. Looking at the singletons, first of all, what you find is that the underfed animals have a reduction in promoter methylation, a decrease in DNA methyltransferase activity, and an increase in GR messenger RNA. Interestingly, the control animals in the twin pregnancies are already reduced to the levels of the underfed singletons. In other words, controls in twin pregnancies that come out from this study already look like stress fetuses, and there's no additional response of the undernutrition in those animals. That's the fetus at term. We then allowed some animals to deliver, to grow up. We came back and looked at the adults of this perturbation at 5 years of age, and what we found was exactly the same changes. When you look up in the top left, decreases in GR promoter methylation in female hypothalami and in male hypothalami, increases in GR mRNA expression, and at the bottom here, increases in glucocorticoid receptor protein as a consequence of a perturbation that's occurred periconceptionally in the mothers of these animals. This is programming, absolutely, I would suggest, of the HP axis in these animals. And if you look at other genes involved with appetite regulation, we saw no differences eventually in females, but we see a reduction in the expression of POMC in the hypothalami of the undernourished animals and an upregulation of NPY. So it's as though these animals have upregulated an appetite-promoting environment in response to undernutrition in the periconceptional period. They've upregulated that environment later on in life. Let me move on to talk just briefly then about the human. Wiley has reminded us many times that in human pregnancy, and Felici and I've discussed this a lot, there's really three HP axes. There's the mother, there's the placenta, and there's the fetus. Over here in the fetus, hypothalamic CRH drives ACTH, and in the human, of course, there's cortisol produced from the transitional zone of the fetal adrenal gland, DHA sulfate from the fetal zone, That cortisol comes back into the placenta. It's hydrolyzed, and glucocorticoids then are one of the factors which upregulate CRH gene expression in the human placenta. CRH, we suggest, then perhaps goes on in increasing amounts through the latter part of gestation, driving processes which lead to term or to preterm birth, including the increases in prostaglandin production. Cortisol from the maternal circulation can indeed cross into the placenta and across into the fetus, but normally, because of this 11-beta-HSD enzyme in the placental syncytial trophoblast layer, cortisol is metabolized in the placenta to the inactive cortisone. And so in normal gestation, only small amounts of maternally derived glucocorticoid reach the placenta or reach the fetal circulation. Does that enzyme activity change in the human? Well, here's data from the Southampton Women's Survey, where we looked at 50 placentae, and the data has never been published other than an abstract because we just weren't sure enough about the way that we interrogated the women. We asked them in a questionnaire format, did you diet to lose weight before the start of your pregnancy? So it's a pretty loosey-goosey question. We didn't know we were going to want to know the answer to it. We had about 20% of women who said yes, they did diet before pregnancy to lose weight, and you can see that just as in our sheep model and as Steve Lie's shown in the rat, there's a very significant reduction in 11-beta-HSD2 messenger RNA and protein in the placenta of those women compared to others. Hypoxia is another very potent factor that reduces expression and activity of the 11-beta-HSD2 enzyme, and that's very likely one of the reasons that 11-beta-HSD2 in the placenta is reduced in response to or in the circumstance of preeclampsia. So infection, low PO2, preeclampsia, undernutrition, steroid hormones, and lipoxygenase metabolites all have effects on this enzyme, most of these reducing the level of that enzyme. That enzyme is also regulated, as we saw earlier today in studies from Vicky Clifton and Isabella Caniggia, in response to changes in fetal sex. So clearly, circumstances in which you reduce the activity of that enzyme allow more glucocorticoid to act on the placenta to activate those stress pathways that are in the placental tissue. Does it matter in the long term? Here's a study that finally Torsten Braun from a charity hospital, Torsten was a postdoc with us, published looking at retrospectively at 44,000 women who were treated with glucocorticoids during pregnancy, showing a very significant reduction in fetal weights of animals, of babies or offspring who received synthetic glucocorticoid, obviously given for pulmonary maturation, but in some of these circumstances given at successive intervals. Does it matter with this, other studies, for example, from Alexander and colleagues published a couple of years or so ago in JCNM, showing very clearly that the responses, particularly of female offspring to stress, tria stress, is much greater in girls from pregnancies whose mums received glucocorticoids during the course of gestation. What are the other things that CRH might do? Well, one of those things, obviously, as Wei Li showed 10 years or so ago now, is to upregulate MMP9 expression in chorion trophoblast cells or in amniotic epithelium cells. In this study here, I'll just go across here, the pro-MMP9 increased in response to CRH. You can abolish that effect, or you can reduce that effect back to basal with antilamin. You see a stimulation out here with urocortin, and again you can reduce those responses with CRH receptor antagonists. CRH and urocortin also upregulate placental aromatase activity, as Alberto Imperatori showed in this collaborative study with Felici, again with the placental either explants, trophoblast cells maintained in culture in the presence of a C19 substrate, androstenedione, and increasing concentrations of urocortin-2, you can see a progressive increase in aromatase activity, a response that's blocked in the presence of the anti-SOVA gene. So what does that mean? Well, it means that these neuropeptides really interact with steroid hormones in the production of in the endocrine responses of the placenta, and as we've heard already today, placental CRH drives the output of dehydroepiandrosterone sulfate from the fetal zone of the fetal adrenal gland. That DS comes back to the trophoblast cells, where, as I've just suggested to you, both urocortin and CRH upregulate aromatase activity and increase estrogen. So again, facilitating a pathway that would be predisposing towards a premature onset of parturition. And if you couple that with activation of a stress pathway, for example, malnutrition, hypoxemia, which upregulates CRH, ACTH, and from the transitional zone, cortisol, driving then more CRH, you can begin to see how these different stress pathways come together in leading to the etiology of preterm birth. So just to summarize this little part, the CRH and the urocortins acting through the appropriate receptor types, uprating estrogens, classically as uterotonins, to increase uterine activation and stimulation, and upregulating expression of MMP9 to promote fetal membrane rupture and cervical ripening. Of course, CRH isn't the only thing that's going to be changing matrix metalloproteinase activity. Here's a study again that Wei Li carried out in collaboration with Al Bocking, where we looked at the effect of LPS, and it won't surprise you that LPS upregulates MMP9. We can abolish that effect pretty much in LPS plus meloxicam, a prostaglandin synthase inhibitor, and then if you hold LPS and meloxicam constant and give back increasing amounts of PGE2 or give back increasing amounts of PGF2 alpha, you can recover the response of MMP9. So the LPS effect on matrix metalloproteinases appears to be exerted, at least in part, through upregulation of prostaglandin biosynthesis, and you can reproduce that with both PGE2 and PGF2 alpha. LPS, as in this study from Felici's lab, Michele Torricelli, showed that lipopolysaccharide added to trophoblast explants increases CRH, urocortin 2 and CRH receptor 1 species, and decreases outputs of urocortin, urocortin 3 and CRH receptor 2. And that's of considerable interest because when you look at the changes in membranes associated with choriamnionitis, you see, in fact, exactly the same thing, that CRH concentrations go up, urocortin 2, CRH receptor 1 goes up, and urocortin 3 and CRH receptor 2 go down, suggesting that the CRH family of peptides, the CRH urocortin family of peptides, have some mediatory role in the inflammatory response that we see associated with infection-driven preterm birth. And as the summary, I don't have time to go through all of these data from Michele and Felici's studies, that the CRH acting through R1 is predominantly pro-inflammatory, urocortin 2 at the bottom acting through CRH receptor 2 is predominantly pro-inflammatory, and urocortin 3 acting through CRH2 are predominantly anti-inflammatory. We've been interested with a very talented PhD student, Mariam Yaganagi, in beginning to try to understand the role of the microbiome in some of these responses. We've been looking at the effects of lactobacilli, and particularly a supernatant fraction that were generated with Gregor Reed from the Lactobacillus rhamnosus GR1 strain. We've shown that in placental trophoblast tissues collected from pregnancies with both a male or a female fetus present, that of course LPS stimulates the output of pro-inflammatory TNF-alpha-1. The lactobacillus supernatant fraction, no effect on its own really on TNF-alpha, but it completely abrogates the stimulatory response we saw with lipopolysaccharide. So this balance that we see between different organisms in the microbiome is a very intensive area of investigation for us. The other aspect here in these data is the suggestion that perhaps the male fetus grows and develops in a more pro-inflammatory environment than does the female fetus, and perhaps that is the root cause of the increases in preterm birth, the predominance of preterm birth in the presence of a male fetus. Grace Yang then took that supernatant fraction and came back and set up the model of LPS-induced preterm birth in mice. Is the receiver operator sort of curve showing you the percentage of animals that delivered in response to LPS alone. GR1 had no effect, GR1 supernatant had no effect by itself, and LPS the effect of LPS is about 50% diminished in the presence of that GR1 supernatant fraction. We're obviously very interested in what is the active component of that fraction, and I don't know at this time, so I'm not hiding anything and saying I can't tell you. What we do know is that that GR1 supernatant fraction is very anti-inflammatory. So if you look at the outputs or the contents in maternal plasma, myometrium, placenta, or amniotic fluid of a number of different cytokines, and just show you some of these in the Bioplex assay. Just focus on TNF-alpha, for example, no effect of GR1 by itself, stimulation with LPS, and in every one of those tissues, that stimulatory response with LPS is abrogated with the supernatant fraction from the lactobacillus. So this the microbiome and the presence, the predominance of the lactobacillus seems to us an extremely important component of this whole story. So let me just summarize the key points I've been making, that preterm birth is high prevalence and cost with substantial regional, ethnic, and gender differences. We don't understand what those differences are. The stuff is just sitting there in the literature, and it seems to me an understanding of these differences is just a field that's absolutely ripe for investigation. So understanding these differences may help understand some of the core mechanisms of preterm birth. Malnutrition, hypoxemia, major stresses that upregulate fetal hypothalamic pituitary adrenal function and result in preterm birth. I hope I've convinced you that the placental 11-beta-hydroxysteroid dehydrogenase 2 enzyme is a critical mediator of placental cortisol transfer and action and of preterm birth and fetal programming. That in women, placental CRH affects placental membrane endocrine function and MMP generation, and the heterogeneous nature of preterm birth requires, to my mind, precision or personalized approaches to diagnosis and to treatment. Thank you very much indeed.